Most nutritional supplementation in the ICU is a benign process. Supplying a patient with L-Arginine operates on a razor-thin clinical tightrope. Administer an arginine-supplemented IV to a severe burn or trauma patient, and the molecule rapidly restores T-cell receptor expression. The immune cycle restarts, accelerating tissue repair and actively saving the patient's life. But administer that exact same IV drip to a patient who has slipped into active septic shock, and the molecule induces fatal refractory vasoplegic shock. Blood pressure plummets to 0. Why does the exact same nutritional substrate cure the trauma patient and kill the septic one? To solve this paradox, we have to stop viewing arginine as a simple protein building block. We have to map it for what it actually is: the body's most volatile and tightly controlled nitrogen asset. In primordial single-celled organisms, free nitrogen was highly toxic. Arginine was selected as a non-toxic, high-density storage reservoir for biologically usable organic nitrogen. This high-density arrangement is made possible by the terminal guanidino group, which contains the most nitrogen-rich side chain of all 20 proteogenic amino acids. With a pKa of 12.48, this side chain remains fully protonated and carries a permanent positive electrical charge across all physiological pH environments. This structural configuration creates an exceptionally hydrophilic center. It facilitates bidentate hydrogen bonds and salt bridges, anchoring the molecule to the negatively charged phosphate backbones of DNA, or the acidic residues of target proteins. This rigid anchoring allows the molecule to serve as the direct catalytic substrate for the enzymes that dictate vascular tone and the limits of leukocyte performance. But constructing this dense arrangement is incredibly difficult. Synthesizing one molecule of arginine from a glutamate precursor costs the cell a net total of 4 high-energy ATP bonds. This high thermodynamic cost and structural density are why the human body strictly rations its supply, meticulously controlling the balance between nitrogen disposal and gaseous signaling. To see how the human body manages this metabolically expensive resource, we have to look at the macro distribution. A healthy 70-kilogram organism contains a total body content of approximately 100 to 120 grams of arginine. The vast majority is integrated into the primary structural peptide chains of myofibrillar proteins and nuclear histones. Arginine is effectively sequestered within the muscle reservoir to stabilize protein and DNA folding patterns. The active transport supply is remarkably small. Only about 10% of the vascular load circulates freely in the plasma, tightly maintained between 50 and 150 micromoles per liter. With a vascular pool that turns over every 1 to 2 hours, the organism is perpetually hours away from systemic depletion if its internal manufacturing network fails. If the body needs to constantly replenish this circulating pool, the logical manufacturer would be the liver, the central metabolic factory. But the liver is a net consumer of arginine. Hepatocytes extract massive portions of portal blood arginine, rapidly redirecting it into the urea cycle to process toxic ammonia. This hepatic clearance is so efficient that if the liver manufactured the systemic supply, its own arginase enzymes would destroy the inventory before it ever reached the systemic circulation. To bypass this hepatic sink, the body utilizes a specialized inter-organ pathway, the intestinal-renal metabolic axis. The intestines manufacture the precursor molecule citrulline. Because the citrulline bypasses the liver entirely, it can travel through the blood directly to the kidneys. The renal proximal tubules extract this circulating citrulline and transform it into newly minted arginine, supplying up to 80% of the active circulating systemic pool. This bypass even relies on the microbiome. Commensal gut bacteria express high-activity urease enzymes to break down waste urea into free ammonia, providing the necessary raw materials for the host to synthesize citrulline. A patient's baseline blood pressure and immune competence rely on this distributed biological black market functioning to bypass the hepatic clearance loops. When that newly minted arginine arrives at a local target cell, it immediately becomes the prize in a fierce microscopic tug-of-war. Competitor 1 is the nitric oxide synthase, or NOS, enzyme family. NOS cleaves the terminal nitrogen to yield gaseous nitric oxide, which diffuses into vascular smooth muscle to regulate vasodilation. Competitor 2 is the arginase enzyme family. Arginase cleaves the exact same molecule into ornithine and urea to drive polyamine synthesis for DNA stabilization and tissue regeneration. Because both systems demand the same limited substrate pool, The cell enforces local compartmentalization to coordinate traffic according to immediate metabolic needs. NOS enzymes are anchored to plasma membrane caveolae. They guard the border, intercepting arginine to instantly transmit vasomotor signals to the surrounding tissue. Arginase 1 is sequestered within the cytoplasm, while arginase 2 is locked inside the mitochondria. They wait for arginine to slip past the membrane to build the polyamine blocks needed for structural repair. This physical distancing of competing enzymes is the sole mechanism keeping the cell from a functional collapse under normal conditions. This cellular architecture explains the ICU paradox. We have to look at what happens during severe burn trauma or advanced sepsis. These hypercatabolic states trigger a cytokine storm, inducing a massive upregulation of inducible NOS across leukocytes and endothelial walls. Simultaneously, damaged cells rupture, dumping large amounts of intracellular arginase 1 directly into the bloodstream. This dual enzyme surge consumes the circulating arginine pool faster than the kidneys can replenish it. Plasma levels plummet, halting the T lymphocyte cell cycle and inducing immune paralysis. Supplementing arginine during active septic shock fuels the iNOS-mediated overproduction of nitric oxide. The resulting wave of vasodilation causes an irreversible drop in blood pressure. Arginine is a direct catalytic substrate for the NOS family. Supplying it during active shock drives the refractory vasoplegia that leads to vascular collapse. To navigate this clinical tightrope, physicians rely on the pathology lab to provide precise measurements of a patient's circulating arginine. Tandem mass spectrometry is the gold standard, identifying arginine based on its parent mass and its unique daughter product ions. This allows the lab to speciate the molecule away from similar isobaric noise. But even the most precise machine is vulnerable to pre-analytical artifacts that create pseudo-hypoargininemia, or fake low results. Erythrocytes and platelets naturally contain active arginase 2 and cell wall transport complexes. If a blood sample sits at room temperature for just 30 minutes, the arginase enzymes inside the tube continue to cleave plasma arginine ex vivo. The measured concentration drops, misrepresenting the patient's true systemic baseline. Mechanical hemolysis creates the opposite error. Red blood cells contain 4 times higher concentrations of free amino acids than plasma. If they rupture during phlebotomy, they spill their contents and cause a falsely elevated reading. Laboratories use automated delta checks to defend against these handling errors, halting the system if a patient's level shifts by more than 50 micromoles per liter within a single day. The most sophisticated mass spectrometer is rendered useless if a simple blood tube is delayed on its way to the centrifuge. Back at the patient bedside, clinicians must recognize one final diagnostic blind spot. This is the normal plasma illusion, the assumption that a healthy plasma concentration rules out local tissue arginine starvation. High metabolic stress induces a localized uncompetitive substrate trap. When arginase 1 is released locally from damaged cells, it aggressively binds and consumes arginine within the immediate intracellular environment of the injured tissue. Because the kidneys continue to release newly synthesized arginine into the renal vein, the circulating plasma levels may remain temporarily supported, even as the local endothelial and immune cells are completely depleted of the substrate. This is why diagnostic consultants must evaluate structural outcomes like wound repair and absolute lymphocyte counts alongside the lab report. You are treating the local tissue architecture, not the isolated number on a printout. The architecture of arginine is built on 3 pillars: the 4-ATP metabolic manufacturing penalty, the spatial tug-of-war between NOS and arginase, and the pre-analytical vulnerabilities of the blood tube. Arginine is the master nitrogen integrator, directly linking a patient's structural nutrition to their systemic vascular survival. Mastering this architecture is what separates standard medicine from advanced diagnostic stewardship. Next time, we explore the precursors in the citrulline salvage networks.